So here we go. So a little bit of a recap here from last time, just to kind of get this whole basic idea, basic chemistry. It was all, everything we were seeing is happening in water, right? That polar molecule that form polar bonds with each other. And also, kind of solubilized things made, was a great solvent because it broke these ions apart, these ionic bonds apart from sodium chloride and other salts so that they're completely dissolved within the solution, right? So now you got these sodium charged ions floating around in the solution, right? If you put this salt in, say, an organic solvent, like, some paint thinner or something, it wouldn't break apart like this, right? So water, polar, important thing. When you put molecules formed by covalent bonds, right? They don't break apart, the covalent bonds, but they are solubilized, right? The individual molecules are kind of separated in these around by water, right? Separating, and so they're also soluble, right? So that's an important point. And then the other important point is that for molecules that are not polar, that don't interact with water, that's another major part of understanding how things act, right? All our cellular machinery and stuff like that, these hydrophobic interactions due to these non-polar molecules, right? That accounts for the formation of the phospholipid bilayer, as well as the shape of proteins and DNA and everything like that, right? So hydrophobic interactions and hydrophilic interactions as well, right? And then those hydrogen bonds, not just with water, but between other molecules, right? Were very important in the molecular shape, which will be a big factor, right? Because one of the things when we talk about homeostasis, right, is controlling pH levels. That'll come up over and over again. One of the reasons we're doing that is really we have to have all like our enzymes and proteins working in optimal form, and they need to do so at a particular pH. Right, so that's just a recap from last time. So for this goes over these it's like one, learning objective, just know the 4 classes, their respective this and this, and functions is really the important part, what we want to talk about today. And then, I put 3 basic functions, just think 2, really, you can cross that off if you're trying to do that question right there. Right? So about these organic molecules, right? Carbon, oxygen, hydrogen, nitrogen, right? Again, just like our man here who is filled up about to the shoulders with water, right? All these molecules are distributed about the body because oxygen is really heavy. It's at the lowest part. Carbon is a little bit, carbon is number 4. This is number 6. So it's a little lighter, and hydrogen is really light. So that's why it's up here, right? Right. No, that's wrong. If I ever say things like that and you start puzzling why I'm saying things like that don't make any sense, I'm just seeing if anybody's paying attention or not. But of course, that's all wrong. This H and O here are making up most of that water, or most of the H and O are coming from that water, and then the rest are coming from these organic compounds, right? So the 4 classes, right? Amino acids, carbohydrates, lipids, nucleotides, right? This is what we're going to talk about. Look a little bit about how they're made and what, their constituent things, what their properties are. But I'm also going to kind of get into where these might come up later, if not in Bio 144, but in Bio 145, right? Understanding the properties might help you understand stuff later on, particularly when it gets to stuff like metabolism. Right? So when we're representing these molecules, when I'm, have pictures of them, you could draw them like, this with all the little carbon molecules in them. Usually for neatness, they're just kind of left off. Every little bend here in this molecule right here is a C, right? And then these other parts right here are like the side groups, right? So you can display these as the bubble space filling model, right? Or these little carbon chains with the little hydrogen molecules. You could leave out those carbon actual C's right here, but that's what this little means. This little squiggly or crooked line means right here. Every one of these bends right here is a carbon. And we always assume, unless there's something attached to it like this, that there's going to be hydrogen in them. Right? There's a couple of different ways of looking at these molecules as we talk about them. And then the other thing is these groups, right over here, like this OH, like this hydroxyl group or this phosphate group, these will be a very important part of these molecules right here, giving them a lot of the properties that we talk about today. So for those, these are the functional groups, right? Hydroxyl, right? All your alcohols are going to contain this hydroxyl group. And then you'll notice also in the description of these right here, I'm going to say things like polar, hydrophilic, right? Molecules with many OH groups dissolve in water, right? This one right over here, this Theos group, polar, hydrophilic, right? So that's an important point. Ketones, right? What else? Polar, hydrophilic, right? This is its interaction in water, right? So all these kind of things you want to notice, like esters and that, but very hydrophilic, right? So all of these are going to give, not only they give a certain charge, maybe in case of phosphate and certain aminos, and they're just going to confer certain properties amongst the molecules, right? On any molecule what we're talking about. So these functional groups that we'll see over and over again. All right, so the next thing is the basic idea of monomers and the subunits of these molecules that we're going to be looking at here, right? So they're always built from single sort of units that are going to be joined together through these dehydration synthesis. That is, you're dehydrating, you're removing a water, or when we don't really talk about this so much in physiology, but we do talk about breaking bonds, right? This hydrolysis reaction, right? Putting a drop of water in here, releasing energy and breaking a covalent bond, right? So again, this is another key aspect of water. Not only is everything happening in water, right, but it's a part of these important reactions. But just to make clear, when we're talking about these water reactions, like breaking apart ionic bonds so that you're separating ions or solubilizing like glucose molecules, that's one thing, right? That's water as a solvent. But this reaction here is the actual breaking of these covalent bonds. So the thing about this is not just water is being added, right? In this case, we could see that there's energy needed to make this synthesis happen, that is joining of two of these molecules together, right? And so you're going to need energy to go in there. And you're also going to need an enzyme, right, to really make this happen, right? Even this one, with this energetically favorable reaction, you're releasing energy. Remember that activation energy hump that you need to overcome. So you're going to need to put a little energy in this to get it. And this is all going to be mediated by enzymes. They're going to bring the water in here, put these two things together, make it energetically unfavorable for a little bit, and cause this to break right here and add the water to these molecules. And that's how we're breaking this apart. So this is very different from solubility, right? This is something enzyme mediated. It's just using water to either, bring two molecules together or break them apart. So this is dehydration synthesis. Dehydration, you're removing water. Hydrolysis, lysis means break. Hydro means water. You're breaking it using water. So our first carbohydrates, right? Carbohydrates, carbon is your carbon background with hydrogen and oxygen in certain places, right? And the ratio is usually two to one, right? Twice as many hydrogen atoms as there is oxygen in these. So any formula you look at, you're going to see this 2-to-1 ratio. And breaking down in their smallest unit, the monosaccharides, disaccharides, and polysaccharides, right? And for our purposes in this class, physiology, we're really interested in the fact that carbohydrates are used as a source of chemical energy. And in particular, glucose, right? The breakdown product of the carbs that we eat, or when we make those glucose, right? We're going to use that glucose energy stored in bonds to produce ATP. Right? So that's our main interest in carbs, right? glucose, right? Whether it's using it in energetic reactions, and we're going to look how it's stored and how it's regulated a little bit, right? For this part of it. So for any carbohydrate, again, dehydration, the hydrolysis, right? We eat sugar, right? We eat a tablespoon of sugar, we eat it, it goes through the digestive system, and somewhere in there, we're going to break those bonds apart to make glucose in this case and fructose, right? If we're eating sucrose, right? So that'll be our monomers here, the monosaccharides coming from a disaccharide, right? And there's a couple other disaccharides here. Our main interest, right, is when we eat starch, spaghetti, potatoes, whatever, those are a bunch of glucose molecules strung together into this polysaccharide, right, to form starch, right, in its various forms, right, starch. And then we eat that, we break it down by this enzyme here and break it down into its constitutive glucose molecules. And that's our interest in glucose right here, right? We don't make sucrose ourselves, right? We don't, so this is what plants do for us. They use the energy from sunlight, et cetera, et cetera. We just take what they make and break it down into glucose. Glucose, over here, this is the molecules, these little positive and negative sort of polar molecules are going to be why it's dissolved in water. So it's dissolved in the blood. It's one of, the main blood sugar is glucose. So it's floating around in there, right? So you've heard of high blood sugar, low blood sugar, right? That's the amount of this free floating glucose in your blood right there. And then for part of the bigger picture here, we're eating stuff, right? You eat stuff, you eat bread or sugar or whatever, right? And your body breaks it down at various places into its constitutive monomers, right? The glucose, that's what's absorbed in your body, right? Once your glucose is in your bloodstream, right, it circulates around the body, right? You can see how long that takes by, if you're hungry or something, you drink, eat something really sweet, candy, ice cream, soda or something, you feel a sugar rush maybe, right? That's how fast it gets to your brain because it's absorbed in there, circulating in the body, and then your brain, which needs a lot of glucose, starts taking it up and uses it for energy. All right, so if you have more than enough glucose, right, you're going to do a couple different things with it, right? It's going to go in through your digestive system into your bloodstream and then pass through the liver, right, which is going to do a couple different things. One of which is just make storage out of it, glycogen. Your muscles will also store glucose as glycogen, right, in there for immediate energy reserves, right? Glucose, there's also a couple other different things that could happen to it, right? Amongst them, turning it into triglycerides eventually and storing it in your fat cells, right? So glucose is this central kind of molecule right here that it's really all about, right? This is where we get all our energy because we need this ATP, right, to do all our functions. So glucose is our main interest in it. As far as this class goes, our main interest in carbohydrates really is this glucose monomer. When your liver gets the glucose, if you have excess glucose, which is, a modern kind of thing, right? Back in the day, if you got glucose in your body did not want to waste it at all, right? It saved everything it got, it reabsorbed it in the kidneys, and it took it all in the liver and or muscles and made it into this glycogen over here, glycogen. And that's just a string in this branched form, right, of glucose molecule. Bunch of glucose monomers right here, all branched and everything. And this is going to be how we store our excess glucose. Just like plants make glucose monomers, make them into starch, a very similar kind of form is this glycogen, right? It's a little bit different, but it's basically the same thing. This is how animals, humans store glucose in this glycogen, right? So this is not to be confused. This is a common kind of confusion point here. Glycerol, right? Gly, right? It's confusing, right? So glycerol, nothing to do with this glucose moniker, this glycogen, but because it's also going to be used as an energy reserve, you might see these interchange. Don't confuse the two, glycogen and glycerol. Glycerol is something totally different, which we'll talk about when we get to lipids. Right? So as far as this making of this glycogen, right, again, when we have too many glucoses, right, this excess glucose, the blood is circulating in the blood. Your blood passes through your pancreas. Your pancreatic islet cells detect those, and they release insulin into the blood, right? So your insulin now is circulating around the blood, and that stimulates. It does actually a couple of different things, but in our interest right here, this insulin is going to stimulate this glycogen formation, right? Both in the liver and in the skeletal muscle fibers, right? Most of your glycogen is going to be immediately stored in your skeletal muscle fibers in the cells themselves, because muscles are high energy intensive. They need a lot of it. And the rest of it is going to be in liver cells, right? So these are the two organs, skeletal muscle cell and liver cells are the ones that can do this formation, this glycogenesis, right? This creation from glucose molecules into the glycogen, right? And so this term is also easily confused with this term glyconeogenesis, which is something very different, which we'll talk about in a second. Right? So that's your glucose storage. Have too much glucose, just put it into a storage form. You don't have a whole lot of glycogen in you, by the way, because it's used up pretty quick. Right? There's only like 500 grams total or something in your body. Don't quote me on that, but there's not that much glycogen in your body right over there. And most of it is stored in your muscles. And so this is glycogenesis, anabolic, making glucose, blood glucose into glycogen. You don't have to know any of this stuff I'm showing you right here. I will mention this molecule right here, this glucose-6-phosphatase, right? But the whole point is blood glucose is used for ATP breakdown, but if you've got enough of it, you need to store it. When you're starved for a blood sugar, when your blood glucose gets too low, this will also, the blood circulating through your pancreas and also other regions will also be stimulated by this low blood sugar. So you have certain other cells in your pancreas that are going to release glucagon, right? And epinephrine is also released by other cells, and they're going to be released in response to low glucose levels, right? So glucagon and epinephrine are going to release that. And then this promotes the breakdown of glucose. So that's a catabolic reaction. You're breaking down that polysaccharide into individual units right here. So this word is not to be confused with this word, which I can pronounce, glycolysis. Right? This one is your normal, that whole in the mitochondria, the glucose breakdown, right, where you're breaking it down, the Krebs cycle and all that stuff like that, right? That's not this. This is something that's breaking down glycogen, not glucose. Right? So that's another common sort of mistake right here, glycogenolysis. That sounds right. All right, so that's glucose storage, glucose release, right? You're breaking it down when you need it, and you're building it up when you store it. So here's the easier picture, right? When you have higher blood glucose levels, right, this is going to get circulating around your pancreas. Pancreas releases insulin, and that's going to cause the liver to take glucose out of the blood and store it as glycogen. Right, so it's going to store right there. Don't worry about this right here. And when you have low blood glucose levels, right, that's also going to pass through your pancreas right here. And that is going to release the hormone glucagon, which is going to cause liver to release glucose into the blood so that it's going to circulate around your blood and get to the body cells. right? So that's your homeostatic sort of connection to this carbohydrate glucose. All right, one thing I want to mention, because this will be important next unit when we talk about membrane transport, things going across according to concentration gradients and stuff, right? Just while we're here talking about this glucose pathway, one of the things when glucose enters a liver cell, a phosphate molecule is tacked onto it right there. And it's not glucose anymore, it's glucose 6-phosphate, right? It's like glucose wearing a little hat or something. And the importance of that is that these receptors right here, these channels that allow glucose in, right, they're doing it based on a concentration gradient, right? So glucose is flowing into the liver cells. And if you have high blood glucose levels, at some point, maybe there's so much glucose that stops coming in. So by putting this little phosphate molecule on the glucose, these channels don't recognize it anymore. So the glucose concentration is always going to be higher out here, because even though there's those glucose molecules, they're not really glucose molecules, right? They're glucose 6-phosphate molecules. Again, don't worry about that right now, but that's just while we're here talking about glucose metabolism a little bit. All right. Here's the other thing. If you are starving for some reason, you have very low glucose level, there's other kind of hormone or endocrine problems with this. But if you need to, you don't have glycogen stores, right? You don't have blood glucose, your blood glucose is too low. You could actually make glucose from these other molecules, proteins, certain amino acids, as well as glycerol, which is going to be part of that fatty acid, as we'll see, as part of the lipids. Right, so you could take either of these and convert them into glucose, right? So this is gluconeogenesis, right? We talked about glycogenesis back then. Right? That was the formation of glycogen. This is actually making new glucose molecules so that can float around the blood, right? Because these other ones don't work directly as fuel, right? They got to be converted into glucose. Right? So when your body runs low on glucose and glycogen, after your glycogen stores have run out, then you start going into this phase. You start breaking down fatty acids and amino acids, right? Which is what that whole fasting trend is to do, right? And ketosis and paleo diets and stuff like that, kind of starve yourself of carbohydrates, and that's going to promote this fat breakdown. There's also other problems with it, but this is gluconeogenesis, right? So you can make glucose from proteins and fats. All right, so that's it for carbohydrates, and next class we'll talk about the lipids.